Welcome back. Today we are discussing a paper entitled VIRMA mediates preferential m6a methylation in 3' UTR and near the stop codon and associates with alternative polyadenylation. This is a paper from 2018 by Yu et al., published in Cell Discovery. In my previous video entitled Transcriptome-Wide m6a Mapping with Nanopore Direct RNA Sequencing, I gave a more comprehensive overview of the m6a methylation modification, which is specifically an RNA modification that impacts many cellular functions, including mRNA splicing, export, stability, and translation. And I recommend checking out the previous video for that more comprehensive introduction. Here we're going to get right into this study where they utilized HeLa cells in order to characterize a new protein that's a part of the m6a methyltransferase complex. In essence, they defined the core complex of proteins characterized to date, or to 2018, as methyl-3, methyl-14, and WTAP, as shown in Figure 1a. They then spend much of the paper characterizing a protein called VIRMA, which they claim induces mRNA m6a methylation in the 3' UTR near the stop codon of proteins, and that a lack of VIRMA leads to 3' UTR lengthening associated with that lost regulation, specifically by somehow mediating or interacting with polyadenylation cleavage factors CpSF5 and CpSF6. This is a study that specifically focuses in on the m6a writer complex as opposed to m6a readers or erasers. m6a, specifically written into the 3' UTR, is known to be installed co-transcriptionally, and it is not a surprise that these proteins therefore interact with a wide variety of transcriptional mediators. But the open question that this study attempts to resolve is, why is m6a accumulated in the 3' UTR and stop codon, and why does it correlate with this alternative polyadenylation? Alternative polyadenylation within the 3' UTR allows for the inclusion of AU-rich regions, microRNA targets, and long non-coding RNA binding sites, which allow for additional complexity in the post-translational or other stages of regulation and modification within the transcriptome. They also report a wide variety of other proteins that are believed to be a part, or that they believe to be a part of the m6a methyltransferase complex. However, these have not been characterized as well to date. In order to specifically generate this figure, they performed a co-immunoprecipitation on these 4 core proteins and then utilized mass spectrometry in order to figure out what proteins were specifically binding to these core components. And then they selected from a list of components that bound to all 3 core components of the methyltransferase complex. They specifically selected these proteins for Western blot analysis and verification, and they noted that most of them associate with WTAP as opposed to the others. They also noted specifically for HNR-NPH that it is related, or that is basically an RNA-dependent interaction. This is repeated in multiple analyses throughout the figures, studying whether an RNA is necessary to mediate the interaction, although they don't necessarily identify what those RNAs specifically are. They next utilized siRNA knockdown in order to figure out whether these particular proteins decreased the level of m6a methylation within polyadenylated RNAs. And they demonstrated that VIRMA showed the largest decrease at approximately 59%. It is also known previously that VIRMA was shown to be an important component of the m6a methyltransferase complex in fruit flies, although its corresponding mammalian ortholog hadn't been studied to date. Interestingly enough, the knockdown of VIRMA siRNA also showed a significant reduction in the amount of WTAP within the proteome. And overexpression ultimately did not recover the m6a level phenotype. So therefore, this implicated VIRMA directly within the methyltransferase complex. Next, this was studied via gene knockout as opposed to gene knockdown or transcript knockdown. And it was found that the VIRMA mutant could only be produced with one mutant allele. Basically, the hypothesis is that VIRMA is necessary for cell viability, and therefore this is the best model. Nevertheless, a significant decrease in m6a methylation was observed within the gene knockout. However, this was not because of a change in any of in the levels of RNA of any of the core components, metal 3, metal 14, or WTAP as measured by qPCR. Instead, it was solely based on this decrease in VIRMA, which was then also shown within this gene knockout system to likely be involved in the stabilization of WTAP, as was observed in the transcript knockdown. Next, in order to better identify what role VIRMA might be playing within the m6a methyltransferase complex, the authors performed ME-RIP-seq in order to characterize m6a levels on a transcriptome-wide basis within both the gene transcript knockdown and gene knockout, and of course within the control HeLa cell lines and knockdown control cell lines. And what they observed is a high overlap within m6a peaks among all of these particular samples of between 60 and 71% overlap. And most interestingly, there was within the VIRMA knockdown and within the gene knockout a significant decrease in modification levels around the stop codon of the 3' UTR. And within the knockdown, a seeming increase within the m6a peaks within the 5' UTR. Generally, however, within the coding region, there was little difference, especially when you look at m6a modifications within so-called long exons, seemingly arbitrarily defined to be greater than nucleotides long, for which there's approximately 80% agreement. On the basis of this, the authors decided to specifically examine further this 3' UTR shift near the stop codon. Additionally, they utilized transcript knockdowns for the core components and performed similar kinds of analysis in order to understand whether this effect is observed with any of the previously characterized proteins. And it did not ultimately seem to these proteins do not exhibit a similar pattern to what was observed with VIRMA. In order to further characterize how this is affecting the transcriptome, the authors examined differential gene sets between the VIRMA mutant and the control, the gene knockout and the control. And they found that m6a abundance inversely correlates to expression pattern. Therefore, greater m6a, less expression, less m6a, as shown here, means more expression. And the gene ontology around this differential gene expression list showed that VIRMA and this regulatory process was seemingly involved with a wide variety of basic cell processes. And in order to verify the discussion around cell proliferation, they looked at the capacity of the mutant cell line to proliferate and found that it proliferated at a much slower rate than normal. They additionally utilized qPCR in order to better verify these dynamics that I mentioned around the inverse correlation between m6a expression and m6a abundance and expression level. So specifically, all 5 of these proteins demonstrated increased expression via qPCR and then decreased levels of m6a abundance. And ultimately, the demonstration within these time decay curves is that the lack of m6a modifications resulted in the transcript not being, as you might call it, discarded of quickly. In essence, the amount of decay was much less. And this is expected because m6a is thought to regulate mRNA decay as one of its many regulatory functions. Next, in order to further characterize how VIRMA specifically interacts with base pair sequences, the authors chose to examine what genes VIRMA binds to, and then specifically whether it binds to the C terminus or the N terminus of the protein, and then basically whether it binds to a specific base pair sequence or not. And so ultimately it's shown that the N terminus is binding to a lot more of these other components within the methyltransferase complex. And that when you utilize specifically the N terminus, so in this experiment, they're utilizing a beta loop and a 5-box B. And in essence, this 5-box B and the beta loop are able to basically artificially bring these things into closer interaction. They attract each other very strongly, and you clone in basically either the N terminus, the C terminus, or other components of the VIRMA complex. And well, in this case, just specifically the N terminus and the C terminus, and you see whether there is more m6a modifications, because you've brought together these 2 known components that are known to interact, and then you have the motif here nearby, and you see whether VIRMA, since it's sort of being forced to interact with this motif, whether that is increasing the amount of m6a methylation. And it was shown specifically that the N terminus was the functional component responsible for increasing m6a methylation and recruiting all of these other components of the complex. And additionally, that it is very, very specific to the classical 5-nucleotide GGACU motif for m6a modifications, as opposed to when you change that motif of GGAUU, you find that this increase in m6a methylation is really abridged to a large extent. Next, a RIP was done. So basically all of the RNA targets of VIRMA were pulled down and intersected with the genes that were known to exhibit m6a modifications in order to see whether there was an enrichment within near the stop codon of the 3' UTR. and this was in fact observed, further verifying the relationship between VIRMA and this specific phenomena. The authors next wish to explore more what was previously given in the introduction as the co-transcriptional nature of these m6a modifications, along with other changes and other regulatory processes occurring during the latter portions of transcription, including splicing and polyadenylation. They utilized a proteomics assay in order to discover which proteins bound to VIRMA, and they found that two specific polyadenylation cleavage factors, CPSF5 and CPSF6, associated with VIRMA in an RNA-dependent manner. Both of these bind near the m6a modification site within the 3' UTR. And as seen in figure 1 and a few other figures, they utilized RNase in order to determine that they did associate in an RNA-dependent manner. Knockdown of these two specific genes did not impact m6a levels, indicating that VIRMA's function in depositing or writing m6a modifications occurred before these polyadenylation cleavage factors act. However, within the VIRMA mutant, it was found that many of the 3' UTRs lengthened based on bioinformatics analysis. Only 13 genes exhibited shortening, UTR shortening. Next, the authors generated a similar gene knockout cell line for methyl-3, which was a part of the core m6a methyltransferase complex, to see whether this could also impact, or whether this had a similar impact on the lengthening of the 3' untranslated region. And it was found that indeed they actually share a pretty similar characteristic, and that there's a significant overlap in the genes that are ultimately lengthened as a part of this regulatory process. The authors then examined whether the knockdown of CPSF5 shortened the 3' UTR of m6a rich mRNAs, and they found that this was indeed the case, as was expected. They then examined whether there was more m6a modifications, whether there were more m6a modifications around the shortened sites here within the knockdown. And they found that indeed there was more m6a modification around these stop codons. And the idea is that these have a higher expression within the CPSF5 knockdown because they lack the polyadenylation regulation. In essence, the components, whether they were the binding sites or the motif-rich regions that would have allowed for regulation, that CPSF5 would have integrated, and therefore either their decay or some other component is not mediated anymore. And they noted that the shortened genes, for the vast majority of them within this knockdown system, are sort of regulated and work together within this m6a complex. And so with m6a and SICPF5, the expression is higher because of, as mentioned, the lack of decay. Next, the paper gestures to potential crosstalk on a mechanistic level between the VIRMA m6a methyltransferase complex and the polyadenylation CPSF5 complex, relying sort of on a difference within the 3' UTR stop codon, within the level of m6a, suggesting some kind of interaction, and also basically a difference between the m6a motif and the proximal APA site, which is where CPSF5 is known to bind, ultimately proposing this summary model for how the two interact in order to produce all of the effects as described within the paper. Thank you very much for listening.